In this video, we will discuss about a novel approach that is proposed in the review article, where they presented an idea that CAR T cell interactions with B cells can be used to enhance its efficacy against the solid tumor. So, before going into their idea, let us quickly revise what this different kind of CAR T cells are based on their differences in the intracellular domain. So, in the top, in the normal CAR has these T cell receptors, and which interacts with the receptors that are present on the cancer cells or any foreign particles, and thereafter it will leads to the activations of the T cells through the intracellular domain CD3 zeta. And what happens in the synthetic first generation CAR, they try to reproduce this T cells activation signaling mechanisms synthetically, where they can change the, instead of using T cell receptors, they use the antigen to target a specific tumors through this variable chain or single chain variable fragment that is shown in this first generation CAR, and it has this hinge to connect it to the intracellular domain, and in the hinge to connect to this transmembrane domain, and it has this intracellular domain that is CD3 zeta, which is used to activate the T cells. So this is the first generation CAR. So in the second generation CAR, instead of just CD3 zeta intracellular this T cells activations domain, they also has the co-stimulatory domain. It will enhance the activations or efficacies of the T cells. It has this one co-stimulatory domain that could be either CD28 or 41BB. In the third generation CAR, they go one step further and included two or both the two co-stimulatory domain. For example, the CD28 41BB, both of them are included. This is the third generation CAR. So these are the different kind of CAR Ts or different generation of CAR Ts based on the differences in the intracellular domain. Now let us come to the idea that is presented in this paper, which they call it as bait and switch CAR T cells. So in this case, what they are suggesting is that to transduce the same T cells with two different CARs. So one CAR has a antigen to detect these B cells, and another CAR has an antigen against the tumors, or they call it as anti-tumor CAR for the antigen against the tumors, and antigen against the CAR they call as anti-B cell CAR. So, and here the B cell CAR that is targeting is the normal B cells, not the tumor or cancerous B cells. So since the normal B cells are present in the human beings, so the CAR T cells will frequently interact with these normal B cells, and this will lead to the activations of the T cells. So the T cells will remain active in the human beings, and these activated T cells can thereafter interact with the or simultaneously can interact with the two solid tumors, could be either in lungs, pancreas, and this will increase the efficacy, since now the T cells has increased lifetime because of its interactions with the B cells. At the same time, they are in the active state. So this will improve the efficacies compared to the case where the CAR T cells has only one, where the T cells only one CAR that is against the solid tumors. So, so compared to that, this approach will provide more efficacies against, or more or better results against the tumor cells or towards this tumor cell killing, compared to the case where single CAR is used. However, one disadvantage of this approach could be the high release of the cytokines. within the body, or cytokine release syndrome that we have discussed when we are discussing the clinical data in our previous videos or in our previous paper. So I hope you understood. So this is just an idea, but in order to make it a reality, there is a lot of optimizations that needs to be done to prevent or minimize the side effects. That's all for this video. I hope you understood this novel idea that is presented in the review paper. If you need to understand these ideas in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.